Okay, now we'll start one by one. First we'll discuss about the parotid gland. So as you know, parotid gland is the largest salivary gland. Okay, so whatever we have discussed, all those things will be applying over here in order to understand the structure of parotid gland. Okay, now in parotid gland, first of all, it is compound tubulo-acinar gland, or also called as racemose gland. Okay, now parotid gland also, it is covered by connective tissue capsule. Now connective tissue capsule will send septa into the gland, which will divide it into lobe and lobules. Same thing. Okay, through that connective tissue septa, blood vessel, nerves and larger ducts will go into the gland. As you can see, blood vessels and interlobular excretory ducts are located into the connective tissue septa. Now each lobule, if you take each lobule, it is made up of cluster of secretory cells where the acini are present. Okay, now you have to know one thing, 3 different types of acini we have discussed, right? Among them, which acini is present in which of the gland, that you have to be very specific. ठीक है, तो जो parotid gland होता है उसमें कौन सा acini रहता है? Only serous acini are present. Okay, जो parotid gland होता है, जो if you take one particular lobule of the parotid gland, then what you will see that it is made up of a special type of acini, and these are only the which type of acini? Serous acini. Okay, now serous acini में क्या रहता है? What are the cells present? What is the duct pattern? Then how to identify? What is the important microscopic features? Everything we have discussed. So everything will be same for the serous acinis. Okay. Now here you can see the cells of the acini, pyramidal-shaped. Spherical nuclei located at the base, slightly basophilic cytoplasm. High power, you can see the granules occupy the apices of the cell of the serous acini, which I have shown you just now. Okay, and here also you can see myoepithelial cells are present. Now the lobules may contain numerous adipose cells also. Okay, so adipose tissues are also present in between the acini. Now the secretions which are taking place, how they will come out? They will come out by a duct system, okay, which is highly branched. So first you can see the smallest duct कौन सा है? Intercalated duct. Now intercalated duct is lined by either simple squamous or low cuboidal epithelium. Next, the striated duct. Now in striated duct already we have seen, just now I have shown you simple columnar epithelium with basal infoldings. That's why striated appearance can be seen. Okay. Next, striated ducts are large, conspicuous in case of parotid gland. Okay, so more number of striated ducts can be seen in the parotid gland, in a section of the parotid gland. Next, the striations are formed. Why? Because of the infoldings of the cell membrane. Now this particular striated duct will empty the product into intralobular excretory duct, which will drain into interlobular duct located in the connective tissue septum. Like this, from one duct to another duct, the saliva is traveling or the saliva is passing. Okay, now as you can see, the lumen of the duct become progressively wider. Okay, as you can see from the intralobular to the interlobular duct, as it passes, the size of the duct increases, along with that the lining epithelium also changes. What are the changes taking place? First of all, the epithelium get taller and increase in the size. Epithelium changes from columnar to pseudostratified, then stratified columnar during the termination. So these are the changes which are taking place in the ducts of the parotid gland. So this is specific for the parotid gland. Okay. Now here is the diagram. This is the schematic diagram, drawable diagram. Okay. Now in this diagram you can see the microscopic structure of parotid gland. ठीक है, तो इसमें क्या दिखाई दे रहा है बोलो? What you can see? No. देखो इसमें connective tissue capsule is not seen, but you can see the septa. Can you see the septa? ये देखो, this is one septa, this is one septa, then this is one septa, this is one septa. Okay, these are the septa. In between the septa, can you see the lobules? ठीक है, एक lobule है. Now take one particular lobule. What you can see inside the lobule? So you have to identify two things over here. Okay, फर्स्ट ऑफ ऑल एज आई हैव मेंशन दिस इज द पेरोटिड ग्लैंड. तो पेरोटिड ग्लैंड में क्या रहता है? ओनली सीरस एसिनस, ओनली सीरस एसिनाई विल बी देयर. तो फॉरगेट अबाउट द म्यूकस एसिनाई, वो नहीं है. तो सीरस एसिनाई का ये क्या है, फीचर्स के ऑलरेडी यू हैव रिटन. First of all, it is darkly stained. Then lumen is narrower. Okay, and then what about the cells? Apical, eosinophilic, basal, basophilic. Okay, these are the things which you can see. So here, can anyone identify which are acini and which are ducts? जैसे देखो ये सब क्या है? What are these? Yes. So what are these? These are the acini. Okay. So this is one acinus, this is one acinus, this is one acinus. Okay. So these are acini. Now what is this? These are duct. But the problem occurs. Student will feel that, most of you will feel that this is lightly stained, so this will be mucous acinus. But ऐसा नहीं है. These are ducts. Okay. Because see, this is lightly stained, this is darkly stained. So appearance, from the appearance you can say that this is mucous acinus, but no, it is not mucous acinus, it is striated duct. Okay, तो always keep that in mind. जो parotid gland है उसमें mucous acinus नहीं रहता है. जो कोई भी अगर dark lightly stained structure आता है तो what do you think? This will be the duct. Okay, जो section में अगर lightly stained section आता है in between the darkly stained section, those are nothing but the ducts. ठीक है, as you can see over here. ये देखो, this is one lightly stained area, this is one duct, this is also one duct. Okay, these are not acini. Okay, so first what you have to identify? You have to identify about the darkly stained acini. Okay, next what you have to identify? Yes, bolo. Next what you have to identify? Next you have to identify about the ducts. Okay, you have to, can you see the duct? So which is the smallest one? Intercalated. Can you see the intercalated duct? ये देखो, this is the intercalated duct. ठीक है. Next you can see the striated duct. As I said, the striated ducts are numerous, or you can say these are more in case of parotid gland. Can you see the striated duct? ये देखो, this is one striated duct. This is one striated duct. Okay, these are all striated ducts. This is also a striated duct. Okay. Now under low magnification, it is very difficult to see the striations of the duct. For that you have to go for high magnification. Can you see the high magnification of the striated duct? ये देखो, this one. Now if you see the basal part, ये देखो basal part, इसमें striations दिखाई दे रहा है, right? So like this you can identify the striated duct, and then you can see the intercalated duct. Okay. This can be identified very easily. इसमें cuboidal epithelium है, इसमें columnar epithelium है with basal striation. Easily I differentiate होता है. ठीक है. Next, I hope you can see the interlobular duct. ये जो large वाला है. Okay. Interlobular duct. So this is one lobule. This is another lobule. In between these two, you can see interlobular duct is present. Can you see the size? Can you see the diameter? Larger. So that can be identified easily. And it is lined by simple columnar epithelium. So this is the way to identify the parotid gland. This is the schematic diagram. This is the original histological image under low magnification. Okay? Can you identify the acini? So which are the acini? Darkly stained. Yes. तो ये देखो, ये acini है, ये भी acini है, ये भी acinus है, ये भी acini. So these are all acini. Can you identify the duct? जैसे ये देखो, ये एक acinus है, but this is a duct. ठीक है. Next you can see ये जो particular ऐसे आ रहा है, these are the connective tissue septa. ये देखो, ये पूरा connective tissue septa है. ये भी connective tissue septa है. These are connective tissue septa. Next you can see this is also a duct. Okay. Now here, though it is under low magnification, that's why it will be very difficult to see the striations. But intercalated and striated duct can be identified. जो intercalated duct होगा उसका diameter कम होगा. You can see the diameter is small. In case of striated duct, the diameter is larger. Next, anyone can you tell me what are these empty spaces? ये क्या है? Adipocytes, okay, adipose tissue. वो भी रहता है. Okay, inside the parotid gland. So this is the way to identify the parotid gland. What is the next one? Submandibular gland. Very good. Now in case of submandibular gland, so previously we have seen parotid gland is a serous acini. Okay, this is not mixed, but jo submandibular gland this is mixed. But you can see serous acini are predominating. इसका मतलब क्या है? What is the more? Yes, the number of serous acini are more. But here you will be getting the mucous acini also. But the number of mucous acini are less. Okay, remaining everything is same like the parotid gland. Duct system is also same. Okay, as you can see, the duct system of the submandibular gland is similar to that of the parotid gland. Now see the diagram, schematic diagram of the submandibular salivary gland. Now here you can see two different types of acini. You can see the serous acini also, you can see the mucous acini, and along with that you can see the demilunes also, serous demilune. Now first please identify the serous acini. Yes. Now submandibular salivary gland, it is a mixed, but mostly you will be getting the serous acini. Can you see the serous acini? ये देखो, ये जो पूरा एरिया है serous acini. This is also serous acini. Okay. Only this particular part you can see mucus as well as mixed acini are present. Okay. Can you see the mucous acinus? So see, this is lightly stained. Okay. These are lightly stained acinus. Here also you can see these are the mucous acinus. Now can anyone show me the demilunes? Yes. ये देखो, this is one demilune, this is one demilune, this is one demilune. Okay, तो उसमें first you have to identify the mucous acinus. इसके ऊपर देखो ऐसे cap shaped रहता है serous acinus. These are called as serous demilune. Next you can see the duct. So duct pattern or the duct system is same like the parotid gland. Okay, जो ducts रहता है in case of submandibular salivary gland same like the parotid gland. But here what you have to identify? You have to identify the serous acinus, you have to identify the mucous acinus, and then you have to identify the demilunes also. Okay. Okay, after submandibular salivary gland, which is the next one? Sublingual. So sublingual is also a mixed type of gland. But what is the difference? Mucous acini are more. Another important difference is there. That is the connective tissue capsule. जो connective tissue capsule होता है in case of sublingual salivary gland, that is not complete, it is incomplete. Okay. So it is a compound mixed tubulo-acinar gland. Now this particular gland is not surrounded by well-defined capsule. That means the capsule is incomplete. Mucous acini are predominantly seen with few serous acini. This also mixed acini are seen, but here you will see large amount of mucous acini. Okay. Here some variations are there. The variations you can see especially in the duct system. Okay, so if you compare the salivary gland, the duct system of the sublingual gland is somehow different. Now how it is different? Intercalated duct are short or sometimes it is absent. Okay. Mostly you will not see any intercalated duct. Intercalated ducts are absent. Next, striated ducts are absent. Instead of that, non-striated intralobular excretory ducts are more prevalent, or more number of non-striated intralobular excretory ducts are present. Okay. Now, interlobular connective tissue septa are abundant in the sublingual salivary gland. Okay, so these are some important identifying feature for the sublingual salivary gland. ठीक है, इसमें intercalated duct बहुत कम रहेगा. In some cases, in some sections, you will not see the intercalated duct. ठीक है. Next, जो striated ducts होता है वो absent होता है. Striated duct नहीं रहता. You will not see any striated duct. Okay. Instead of that, you can see the intralobular excretory duct, that also non-striated. Now this is the diagram. What you can see over here? Yes, so you can see mostly the mucous acini. तो ये देखो, ये पूरा mucous acini है. This is also mucous acini. Along with that, you can see few serous acini. Okay, ये देखो. This is serous acini, this is serous acinus, this is serous acinus. Only few. Okay. Rest you can see the demilune. So ये देखो, this is one demilune. Okay. Here also one demilune. Next, here you can't see any striated duct. Okay. Instead of that, you can see intralobular excretory duct. Okay, so mostly you can see these are the mucous acinus. Here also mucus. Along with that, only few serous acini and demilunes can be seen. Okay. Next, as I said, the most important part for the duct system is striated ducts are absent. Even intercalated ducts are very less. Sometimes it is absent. Okay. Apart from this, you can see non-striated intralobular excretory duct and larger interlobular excretory duct are seen. Now, these particular ducts will be present in the connective tissue septa. Okay. So, that is all about the sublingual salivary gland. Okay. Next topic is tonsil. Okay. Now, what exactly is the tonsil? Before going to the microscopic structure, you should know what exactly is the tonsil. Okay. Where it is present, what is the important function, and how these tonsils are formed. Okay. Now, what do you think where the tonsils are present? No. This is not the proper definition. Okay, this is not the proper definition. That's, I know most of the student will be having difficulty. That's why I have added this particular part and a diagram to show you the exact location of the tonsil. Okay, and that will be discussed in the gross anatomy also. Don't worry. So near the junction of the oral cavity with the pharynx. Okay, so जो oral cavity है and pharynx है, उसका जो junction होता है near that, you will be getting huge amount of aggregation of lymphoid tissue. Okay, now that aggregation of lymphoid tissue is in the form of a ring, or you can say in the form of a circle. Okay, and that is called as Welder ring. So the junction of the oral cavity with the pharynx, number of collection of lymphoid tissues are there, and those are called as tonsil. Among them, the largest one is the palatine tonsil, or also referred to as tonsil, which you have shown just now. Okay, when I ask you about the tonsil, you have shown this one. Whichever you have shown, that is called as palatine tonsil, the largest one. Okay. Next, some other parts are also there where these tonsils are present. For example, on the posterior wall of the pharynx, that is called as pharyngeal tonsil. Then on the posterior part of the tongue, that is called as lingual tonsil. Above the tubal opening of the auditory tube, another tonsil is present. These are called as tubal tonsils. Okay. So all those aggregation of the lymphoid tissue, if you take all the aggregation, you can draw a circle, and this is bilateral. मतलब इधर भी रहता है, उधर भी रहता है. Okay, so you can draw a circle. If you draw a circle, it will appear as a ring, and that is called as Welder's ring. Okay, this is called as Welder's ring. Now here is the diagram. Okay. Now in this diagram, can you see the palatine tonsil? This is the palatine tonsil. Okay. On the either side of the oropharyngeal isthmus, we will study what is oropharyngeal isthmus. Don't worry in gross anatomy. On the either side of the oropharyngeal isthmus, you can see the tonsillar fossa is there, where this palatine tonsil are present on either side, bilateral. Next, this is the opening of the auditory tube. So you can see above the opening of the auditory tube, aggregation of lymphoid tissues are there, and these are called as tubal tonsil. Next, on the posterior wall of the pharynx, you can see pharyngeal tonsil is present, and on the posterior aspect of the tongue, lingual tonsil is present. Okay. About the tonsil, we will be discussing mainly about the palatine tonsil. Okay, the structure of palatine tonsil. Very simple structure. So the palatine tonsil is made up of first lining epithelium is there. Okay, the lining epithelium is same like the lining epithelium of oral cavity. It is stratified squamous non-keratinized epithelium. Okay. Next, you can see the tonsillar crypts, that is dipping down or invagination of the lining epithelium down into the gland, down into the substance of the tissue, that is the tonsillar crypts. Now the crypts are also lined by the same epithelium. Next to the lining epithelium, we are getting the lymphoid tissue, where we will get the aggregation of lymphatic follicles and those are supported by the reticular fibers. So that is the overall structure of palatine tonsil. Okay. Now the boundary between the lining epithelium and the aggregation of lymphoid tissue, okay, as such boundaries are not there. Okay. Next, what is the function? Production of activated lymphocytes and antigen presentation and development of the immune response. So these tonsils, if you see the location of the tonsil, okay, they are situated in an area where it is encountered by the air which you are breathing and food whatever you are swallowing, and thus they provide the first line of defense to our body. Okay, because mostly pathogen enters in our body through inhalation, because every time you are inhaling, right? Every time you are inhaling, so whenever you are inhaling, the air has to pass through that only, oropharynx only. So in that particular part, this aggregation of lymphoid tissues are present. So they are providing first line of defense to our body against any pathogen which are entering in the body. Okay, that's why this tonsil is very important. Now there are three parts. If you take a section of the tonsil, three parts can be seen. First of all, lining epithelium, tonsillar crypts and lymphatic follicles. So what is the lining epithelium? Stratified squamous non-keratinized epithelium. Then you are getting the tonsillar crypts. I hope you remember the intestinal crypts when we have discussed about the intestine. Similarly, here also crypts are present. Okay, जो crypts रहता है उसमें भी same lining epithelium present होता है. Next, lymphoid tissue just beneath the epithelium, palatine tonsil shows dense segregation of lymphatic follicles, and those lymphatic follicles are supported by reticular fibers. Now here you can see the diagram. Can you identify the lining epithelium, anyone? Yes, can you identify the lining epithelium? ये क्या है? What is this? Lining epithelium. Very good. Then what is this? Crypts, tonsillar crypts. ठीक है? Now crypts में भी देखो same epithelium है. Now if you see the below the epithelium, ये lining epithelium, इसके नीचे ये क्या है? Aggregation of lymphatic follicle. Can you see the round structure? ये देखो, this round structure. These are the lymphatic follicles. Okay? Lymphatic follicles भी दो टाइप का होता है, primary lymphatic follicles and secondary lymphatic follicle. प्राइमरी में जर्मिनल सेंटर नहीं रहता है, सेकेंडरी में जर्मिनल सेंटर्स रहता है. ओके, एंड दिस lymphatic follicles, these are supported by reticular fibers. Okay, and along with that you can also see the blood vessels. Okay, so previous diagram, this is important. Okay, this is important. Okay, this you have to draw, this particular diagram you have to draw in a practical notebook. ठीक है,